Okay, I want to talk to you a little bit about some of the work from my lab where we studied the yeast replisome at the single molecule level. And a few years ago it became possible to really fully reconstitute the yeast replisome by chemical experiments. And so the main ingredients of what it takes to establish a replisome are known. We start with a so-called origin of recognition protein shown here, and several others in the first phase of this loading phase of DNA replication, where the goal is to establish the double hexamer of MCM, which is an inactive form of the helicase. There will not be a quiz afterwards, asking you about all the different components of the replisome, because there are indeed many more. But I'm just going to say that you need a number of other factors, order the 10, to sort of get yourself to the mature form of the helicase, which is called CMG, where the M in CMG stands for MCM, which is, again, right, this inactive form, and the C will come up later because it's one of the other proteins that we label, CDC45. This active helicase is then competent for unwinding the DNA, which is, of course, necessary, right, prior to replication, and then you essentially need to add the polymerases that will then synthesize the new nucleotide and add them so that you can make yourself the daughter strands of DNA. So as I mentioned, the biochemists managed to make this work in the test tube in 2015, in particular John Diffley's lab at the Crick, and since then my group has been collaborating together with him in order to establish this system and its dynamics at the single molecule level. Okay, so my lab is a single molecule lab, and so when we realized that you could do that, the biochemistry to some extent was clearly working, we thought it would be very nice to look at the dynamics of these proteins using single molecules. And a question that we had to deal with was kind of what is the right single molecule approach for this type of complex system. So my lab had done a lot of work using magnetic tweezers, where you tether DNA to magnetic beads and pull on them using magnets, as shown. And the readout here is the position of the magnetic bead, and it has the huge advantage that you can have many of these magnetic beads, each with their own DNA tethered to them. But as the readout is the magnetic bead, if you're adding, let's say, 15 different types of proteins, it's kind of hard to know what's going on if you only have that the bead motion as the readout. So we rejected this. So then we thought, okay, people had done very nice work also on viral replication using turf microscopy. So we thought, let's do that, where you have tethered DNA molecules along the surface, and then you use turf, as we heard about in one of the earlier talks, and you image, right, the different proteins. This was also, well, has the advantages of being high throughput, but to us had the disadvantage of having the surface in proximity, right? If you're trying to set up a protein complex and you need to add proteins in several different steps, there's lots of potential for sticking, and that was realized in our experiments. So, of course, and then there's also another technique that's known in the field where you called optical tweezers, where you can hold your DNA fully away from the surface, as shown, and typically people would study proteins by tethering a protein to the bead and having them pull DNA through, et cetera, et cetera. Also very nice, high resolution, but low throughput, and not so useful in studying replication if you also need to visualize the protein. So we end up with this approach where you have two beads holding the DNA, and then a scanning confocal laser that allows you to image the proteins, as indicated here. And for us, the main advantage of this technique in our experiments is that the optical traps lift the DNA away from the surface. As mentioned, the confocal scanning laser allows for visualization, but the drawback is that we go back to the early days of single molecule techniques, namely in the fact that the throughput is low. We do these experiments in two different ways. You essentially assemble a tether, and then we can either go into a channel that has labeled protein and then have protein bind, exit, and image, or alternatively, we can essentially flush in fully formed DNA protein complexes into the instrument and then image those. And I will use both modes in what I'm going to tell you today. So the first part is the loading phase of DNA replication. As I mentioned, there's this origin recognition protein.
There's an intermediate that I'm not going to say so much about, and then ultimately the goal is this double hexamer formation. So let me tell you a little bit about what we could see using the ORC protein labeled in green. Right, so the way we do the experiments is that we take our DNA and dip it into this channel with labeled ORC for 2 minutes, and then we go to a separate channel where there's no free ORC, and we image it. And we see a bright green spot as indicated here, and then you have the beads, which also have some DNA and protein on them on either edges of the scan. And from biochemical experiments, it's known that the ORC is not stable to high salt. So if we do a high salt wash, it should go away, and it does. And this is with the yeast ORC. So we have it will recognize in biochemical experiments and shown it recognizes a particular origin, and the origin is indicated here in red. Then we can also say, if we repeat this experiment many times, where is the ORC protein bound? And that's shown over here. Now, in these experiments, you don't control the orientation of the DNA. So essentially, you need to be able to plot distance from the DNA center to symmetrize your data. So here, the origin is at this double dashed line, and you can see that ORC binds at many positions on the DNA, but it does preferentially reside at the origin. And you can do things like count the number of ORCs and monitor the stoichiometry and so on and so forth. So what's also known from biochemical experiments, in particular EM, is that the ORC will bind to the DNA when it's bound to it. And that means that if that's trying, if you're essentially applying a force during this incubation period, you would expect the binding of ORC to be sensitive to the force that's applied to the DNA. So that's the experiment that we did here. So we plot essentially either the fraction of foci that have the fraction of DNAs that have an ORC foci at the origin as a force, or conversely, the number of empty DNAs as a function of force. And you can see that indeed, as the force increases, that the presence of ORC decreases, because of course it would cost more energy for ORC to bind the DNA. So these findings are in single molecule, essentially confirming what's known biochemically. And the last kind of control that I have here is that if you mutate the origin, then biochemical experiments will indicate that ORC will bind less frequently, and that's also what you see in this spatial distribution plot using a mutated origin on our long DNAs. Okay, so then of course the main interest of doing these single molecule experiments is that you can look at their dynamics, and that's what we do here. So what we've plotted here is the DNAs as we image them in the setup. So here the data is not yet symmetrized, and we plot the location of ORC as a function of time. And in this plot, what we've said, okay, here at t equals 0, we say, where is the ORC? And we say, and these are the traces that are filtered based on finding ORC in a bin close to the origin at t equals 0. And you can see that most of these molecules essentially stay at the origin. If you do the same thing for other traces where ORC started somewhere else in the initial frame, then you can you get the data that's shown here in the center, and you can see by eye that these traces are more dynamic, right? And that's also, this is just a movie version showing you that ORC is indeed not static on the DNA, but essentially scanning it. And in general, we find 4 kinds of traces. We find either ORC initially bound close to the origin and staying there. We find ORC initially binding somewhere else along the DNA and being mobile. Or we find case 3, ORC initially bound close to the origin, but able to leave. Or case 4, ORC binding somewhere else initially on the DNA, traveling along the DNA and locating the origin and staying there. So there's the 4 general characteristics. But those are just traces, right? And so as biophysicists, we like to quantify and convince you that this is all statistically sound. So we can do that. So per trace, what we've done here is calculate a diffusion coefficient for ORC on our standard DNA molecule, and you can, this is plotted here in log, but what I want you to appreciate is that we have a population of fast moving ORC molecules and a population that we just call slow, where slow can also mean static. This is 1 diffusion constant per trace. So the molecules that are somewhat static and somewhat mobile will give some kind of intermediate diffusion coefficient. Stable and static, of course, in the DNA. That essentially gives you a measure of, what's our best estimate of static? How slowly moving the molecule can we really measure? And then another control, the DNA with a mutated origin. And you can see here that what we essentially lose all of the slow the molecules, and we are only left largely with the fast moving ones. And then another form of quantification. So we say, how many molecules of the ORC end up being quantified as being part of the slow population, depending on the sequence at which they start at. So here, this is for different biochemical conditions that I won't elaborate on, but essentially if the molecules start at the origin, they tend to, roughly half of them end up being classified as being slow. There's origin-like sequences, and then you can see that this fraction goes down, and then there's essentially everything else, and then you can see that the fraction of ORC molecules that end up being classified as slow essentially goes to only sort of 10%. So that clearly shows that we have a correlation, of course, between ORC mobility and sequence. And you end up with this picture, of course, where ORC is essentially binding initially aspecifically to the DNA, diffusing along the DNA, and upon recognition of the origin sequence, likely to bind there stably. So that's my story about ORC. Then, of course, as I mentioned, the ultimate goal of this loading is to establish the MCM double hexamer. I'm going to skip the intermediates. Here we do the experiment slightly differently. We do things in the test tube initially, then put it in the single molecule setup, and then we may perform a high salt wash to see what is really stably bound to the DNA. And the main thing that I want to mention about that is if you do the reaction in ATP gamma S, biochemical experiments will say that the products are not salt stable, which is what we confirm here. And then if you do it in ATP, then they are. In particular, the MCM proteins are then stable on the DNA. So that's what our single molecule experiments confirm. And just like the ORC measurements, what we can then do is spatial positioning quantification and stoichiometry quantification. In pink, single hexamers, right? And in red, double hexamers. The main thing here is that these are broad distributions, but they're roughly centered about the origin. And if we quantify stoichiometry, it's either largely 1 or 2. We were glad to see 2s, right? Because the ultimate product of the reaction, let's say the biochemically interesting project, is double hexamers, which we do find. And then one of the other questions that we had was, what is the mobility of these MCM proteins? From biochemical experiments, it's known that it's possible to move a double hexamer, that it can diffuse on the DNA. But so we tried to quantify that in the same way that we did for ORC. And this is for single hexamers and for doubles. Very little was actually known about single hexamers and whether they were mobile at all. Initially, we thought, well, this doesn't look very mobile. It looked actually rather similar to DCAS9. So essentially, it's not a very mobile protein. So then we said, okay, let's do the experiment again, but just have very long time intervals between frames, right, from 0.6 seconds to 120. And then you can indeed compare the mobility of the MCMs, either single or double hexamer, to DCAS9. You can see that indeed it is a mobile protein. So right, in terms of the biophysics, you can get a real diffusion constant for MCM. And what's interesting in terms of the biochemistry is that you can really establish that also the single hexamers are indeed mobile proteins on the DNA. And this has implications for the role of establishing the double hexamer, right? To what extent does a mobile single hexamer play an intermediate? There's a lot of discussion about that in the field. And then these MCM proteins are also useful as people are finding and play roles in processes other than only sort of standard replication alone, and having some mobility there may also be biologically interesting. So that's what we learned essentially about the loading phase. And what I want to now go on to is essentially the next step, right? I said the next step is to form the active helicase that can really unwind the DNA. And I alluded to the fact that you need to add a lot of different proteins. And we found certain that we could do this in our single molecule setup, but then it caused some problems for the DNA, got a little too crowded, got a little too sticky, etc. So we made a hybrid protocol where we first do the biochemical reaction bulk on beads, and then we wash the complexes, which you always have to do, otherwise you have too much aspecific binding, and then we put this back into the single molecule setup and then image what's there. And this, this means my students do essentially a morning of biochemistry before doing single molecule experiments in the afternoon. And hopefully we catch then the CMG helicase. So we have a label on CDC45, which is the C in CMG. And we again see a green spot. So it's a kind of a coincidence that both ORC and CDC was labeled in green, and now I also have a green protein, but this is CDC45. And we like to be sure of what we see. So we look at the number of spots that we see per DNA, which is typically 1, but sometimes 2, the number of CMGs per spot, which is a distribution shown over here, and then what gives you ultimately the total number of these CMG helicases per DNA. So it's mostly 1 and sometimes 2, essentially. You could argue, if you looked closely at my diagrams, you could see that we start with a double hexamer, which should evolve into 2 CMGs. And we start the reaction essentially in bulk, so some of them may already be moving, right? So you would say, well, okay, you should always have multiples of 2 CMGs, and obviously 1 is not a multiple of 2. So why is that? Well, we don't know for sure, right? But okay, we have to do some handling. Of course, in these experiments, we do this high salt wash, and then we have to put it into the single molecule instrument. The CMGs can be mobile, and if they then encounter a nick on the leading strand, essentially they can fall off. And then possibly may also be the case that sometimes only a single one of the 2 MCMs may mature into a CMG. This has also been seen in other experiments. Okay, so then we go through, so hopefully I convince you that we see some CMG here. Again, we can also quantify where are they located. And this is split here into location as a function of stoichiometry. But again, as with the MCMs, the main thing is a broad distribution centered about the origin. And so we can, that's consistent. It should be centered about the origin. That is where they should normally start from. And then we also, again, try and touch base with what's known biochemically. So we know that it should be salt stable, which we verify. And then it should depend, this is a factor I didn't mention very specifically, but the formation of the CMG is critically dependent on this phosphorylate, this kinase that phosphorylates MCM, which is called DDK. And we wanted to convince ourselves that that was the case, which we did. And here I show you the data for that. So here what we have done is we've labeled MCM in red and CDC45 in green. And what you should see, right, is that you should see more CDC45 in an experiment where DDK has been added. So that's this one, plus DDK, minus DDK over here. You can see that there's more CDC45 in this experiment than in this experiment. And additionally, you should convince yourself that these two are frequently co-localized, right? So we know that we can load free MCMs, as my earlier data showed, but if we're really forming CMGs, there should be MCMs that are co-localized with CDC45, and so that's what we quantify over here, right? The fraction of MCMs that are co-localized with CDC45 in the presence or absence of DDK. So this is clearly it's much more favorable in the presence of DDK. So this is a proper biophysical manifestation, right, of what the biochemists know. That means we can then start to actually do, right, the biophysics using in the single molecule experiments. And as in the case of ORC and MCM, we have essentially looked at mobility. Again, what is mobile? So this is again the DNA molecule as a function of time. Here we've bound dCas9 to the DNA, and so that gives us a measure of, right? Obviously, we have some noise in the instrument, so we can quantify that and sort of say, okay, below a certain limit, we know things that has to be static. So that's essentially a velocity distribution that we get for dCas9, essentially reflecting the noise in the instrument. And in our single molecule data on CMG, right, we essentially say, anything you need to have a change point segment that's 5 sigma beyond the width of this dCas9 distribution for us to consider any trace with CMG to be mobile. So let me now show you what the data actually looks like of moving CMG. I have a movie here first. So again, CDC45 in the CMG in green. So you can see several of them moving every now and then. Well, every now and then, rather infrequently, we catch it very at the start of the reaction, right? The two CMGs really starting starting as two and moving apart. That's infrequent because, as I said, we trigger the reaction in bulk. But nonetheless we see it, and then of course we can also quantify all kinds of other traces that we take. So motion of CMGs along DNA in all these different traces as a function of time. So this, you can see some of the fits here using change point analysis. And we can also quantify based on this data the number of static traces, which for DCAS9 is, of course, essentially 100%. And for CMG in the presence of ATP goes down to about 30%, meaning about 70% of the traces show mobility. We were a little bit surprised when we did this control experiment where we didn't add ATP. We thought, okay, it's just not going to move. Okay, but this is not correct. So this is actually what we see. We see that CMG is also mobile in the absence of ATP, and perhaps you can see that its behavior looks also to be a little bit more random just from this movie. But if not, you can see it from the traces here, right? So position along the DNA as a function of time in the absence of ATP for CMG. And so it looks more diffusive, but okay, first I quantify also the number of static traces is kind of intermediate between ATP and no nucleotide, but that still means 40% of the traces show mobility. Okay, so it looks different. Okay, let's quantify, let's convince ourselves. So we have two ways of doing it. So the first way that one of the people in my lab devised was to say, okay, well, if I have unidirectional translocation, then my change point segments should, by and large, consecutive segments should point in the same direction. Whereas if I have diffusive motion, right, on average, only 50% of the traces should then point in the same direction. And this is what we've analyzed. This is the case of ATP. Nearly 100% of the segments have the same slope, same sign of the slope. So that's unidirectional translocation. And then in the absence of ATP, you can see it's only 50%. So suggesting diffusive motion. That's one way. The other way is that you really do MSD analysis, right? So looking at mean square displacement as a function of time and fitting it. From that, we can get right diffusion, well, exponents of this behavior. And so we can quantify things as unidirectional diffusive or subdiffusive. In ATP, you can see the primary population is unidirectional. Without ATP, you can see the primary population is diffusive. Nonetheless, you can see there's also some say, additional populations in both of these plots. And we thought, okay, do we need to interpret this biochemically, or is this a result of the analysis that we do or the experiments that we do? So we did a simulation using similar parameters and similar trace lengths is what we gather experimentally, and then we refitted those using the exact same analysis algorithm. And then you can see that essentially the simulations give you nearly identical results to the experiments, right? So that means that, for instance, in this case, the fact that you see some traces that are quantified as diffusive, those are probably very short, relatively short traces, where it was just, yeah, it was, if it had the trace lasted longer, it would most likely have been classified as ultimately being unidirectional. So from this, we conclude that in the presence of ATP, CMG preferentially moves unidirectionally, whereas in the absence of ATP, it preferentially moves diffusively. And then just sort of summarizing this, right? So this I just said, but in ATP, we also see some statics, and then without ATP, we see in terms of motion diffusive and also some statics. So let me just try and put this into a working model for you. So this is, again, relying on some things that are known from the biochemistry. So it's known that CMG can slightly melt the DNA already, and that this step requires binding of nucleotide, binding of ATP. And then there's another step, which requires ATP hydrolysis and an additional protein factor called MCM10. And in that sort of makes it possible for the CMG to surround only one of the two strands, right, in preparation for the unwinding of DNA. So this is known from biochemistry. So what we presume is the following, right? So we do our experiment initially in bulk, right? In ensemble measurement, we don't see what's going on, right? And so presumably we have a combination of these two states. And then we wash with high salt, etc., and put it in the single molecule instrument. So during the high salt wash, we remove the nucleotide, and then we add some form of nucleotide back or not, right, in the single molecule experiment. And then we get these outcomes, and let me just guide you through them. So if, right, there was a CMG already surrounding DNA in a single strand of DNA, and we give it ATP, it's probably just going to unwind the DNA, right, unidirectional motion. And if we give it no energy source, it's probably just going to stay there, right? So presumably this will be static. Now, we know that in the absence of ATP, we also see diffusive motion. So what we assume is that these derive from this melted state. Then we remove the nucleotide, right? So probably it's diffusive here already. We don't add anything more, so it just stays diffusive. And then lastly, we can, if we remove the nucleotide here and then we add back ATP in the extruded states, it can't start unwinding because it's not in the extruded states. So presumably it's just this accounts for the static population that we also see in the case of ATP. So a prediction that this model makes is that, for instance, if we were to, that essentially nucleotide binding can halt a diffusing molecule, and we wanted to check that by also performing the experiment with ATP gamma S, right? In that case, we should really see only static molecules. So that's the last single molecule experiment that we did, and you can see the results here, and you can see that the overwhelming majority of molecules is static, which is also what you see in this bar plot here on the right. So this essentially implies that without the nucleotide, CMG can diffuse on the DNA, and that the binding of nucleotides will halt it. So that brings me then also to my conclusions, which I have summarized as follows. So we worked on the loading pathway of reconstituted yeast replication on these single molecules of DNA. And what I want to emphasize is that we looked at mobility and we related it to different factors, right? So in the case of the ORC, we related it to the sequence of the DNA. And then for the case of the MCM, the inactive helicase, we linked it to stoichiometry. It turned out didn't matter what the stoichiometry was, right? They were both mobile, but we did examine. We were able to examine exactly that, whether there was an effect or not. And then, well, anyway, okay. So then I think my things are out of order here. Yeah. So we have made this crossover assay to look at sort of a more complex step, right? Where more proteins involved, where we do ensemble biochemistry with single molecule biophysics, and that allows us to image fully reconstituted CMGs and their motion for the first time. And then again, we look at mobility, but now we relate it to the nucleotide state, right? So in the presence of ATP, CMG moves unidirectionally. In the absence of ATP, it moves diffusively. And then nucleotide binding can halt the motion. Well, it can halt the diffusive motion of the CMG. Yeah, I think it's nice that, right, you can do biophysics to look at mobility and link it to all these different parameters. And I think, excuse me, oops, well, I'm going the wrong direction. Make it up here. Yeah, essentially what's nice is there's plenty of beautiful biochemistry that's done in the field and also very nice EM. But with these single molecule experiments, we can really shed light on the protein dynamics during the loading and activation phases of DNA replication. And I just want to thank a lot of people in my lab. They're all listed on the left. And I want to thank John Diffley for collaboration on this work, and Alessandro Costa and Jacob Lewis for reagents on the CMG part of this work. Thank you for your attention.
Thank you. Thank you very much. Wonderful talk. I was wondering what would be the role of centromeric chromatin proteins in such kind of movement, especially when you're talking about arcs, autonomously replicating regions, or for example. So what would be your speculation on the role of centromeric proteins?
Oh, well, let's say, I guess we certainly have done no experiment like that, but of course it has a lot of repetitive DNA. And certainly people have shown in bulk that a lot of repetitive DNA can stall the replisome. It depends a bit on what kind of repeat it is and how many repeats there are. And I don't know whether, I mean, it's Gidon Koster who's done a lot of this work recently, very nice. He's in London, and he has certainly shown that it, like I said, depends on the type of repeat and the number of repeats, but whether one of the ones that he was testing was a repeat that occurs frequently in centromeric DNA, I wouldn't be able to tell
Sure.
you.
Thank you.
Yeah.
So the question online is it possible to do these experiments changing the tension of the DNA template?
Yeah. Okay. Yeah, that's a fair question. So I showed you one experiment where we changed the tension, right, when we were incubating with ORC, and then we could see an effect. By and large, we have not changed otherwise the tension during the actual imaging. And the reason for that is that, okay, we don't want to have the tension to be too high, because then we may be stretching, well, we will be stretching the backbone of the DNA, and it's not very clear what that will do to the normal mode of operation, right, of these proteins, which typically will find DNA in less tense situations, let's put it that way. And then we also don't want to have the force be too low, because then you decrease your resolution during the imaging. So we have largely done these experiments at 2 piconewtons. We recently did a CMG experiment at a higher force. And the reason that we did that experiment is that there was mention in the field that CMG could be diffusive on DNA in an experiment where first the DNA was subjected to 65 piconewtons and then returned to a force of 10 piconewtons for imaging. So it was diffusive in the presence of nucleotide, right? And that's not what we saw at 2 piconewtons. So as a control experiment, we repeated it at 10, and we still do not see diffusive motion of the CMG there. So it's useful, of course, to be able to change the force. If anyone's interested, I can chat with you about why I think this other experiment probably does not reflect physiological behavior of CMG.